Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Plants use a variety of mechanisms to communicate with other organisms, including one another. Volatile compounds can signal flowering and attract pollinators, for instance, and mycorrhizal fungal networks can transmit warnings or transfer resources. Small RNAs are on that list of communication molecules, and new findings confirm their potential. According to a paper published October 14 in Nature Plants, the plant Arabidopsis thaliana secretes microRNAs, mRNAs, a type of small, single-stranded RNAs, into its liquid growth medium. In addition to their role as regulators of gene expression within an individual, as part of development or in response to stress, they have been implicated in defense against pathogens in recent years. For instance, Arabidopsis cells infected with the pathogenic fungus Botrytis cinerea secrete small RNAs packed in extracellular vesicles that, when delivered into their attacker, inhibit its virulence. Plants are also able to take up sprayed RNA molecules targeting genes from pathogens. The recent findings are the first evidence of plants taking up RNA secreted by other plants into the environment. As they simply wanted them to produce seeds, he adds, the researchers didn't care about placing different plant lines in separate trays. But then they noticed that wild-type plants sharing the mutant's hydroponic solution had phenotypes different from those expected, for example, those growing next. Furthermore, cultivating both lines in the same solution resulted in wild-type plants with notably lower expression levels of the genes targeted by the mutant's boosted mRNA molecules. One possibility, Parata posits, is that sharing information by exchanging RNA would allow plants experiencing a stress to warn nearby plants, not yet affected by the stress. Competition could be another explanation, he writes. For instance, if a plant releasing mRNAs could inhibit physiological. However, the researchers found that applying extracted, presumably naked mRNAs or synthetic RNAs had an effect in gene expression, suggesting that exosomes aren't needed for uptake. She suggests in an email to the scientist that, as with nutrients, plants might actively assimilate small RNAs from the environment. But unlike the substances plants are known to import, naked RNA molecules were thought unstable, she says, so RNA uptake was ignored or underestimated. See, exosomes make their debut in plant research. Jin agrees that the evidence in the paper supports the hypothesis that plants can uptake Na. She adds she also suspects that these vesicles could protect the mRNAs, helping the plants to accomplish a more efficient uptake. Otherwise, the molecules could be more easily degraded in the soil and in the environment, she speculates. Guo points out that, as this mechanism has only been explored in hydroponically grown plants, it's not yet clear whether seedlings growing in soil would have effects on regulation of gene expression in nearby plants, something future studies could examine. Jin adds that these new findings open a lot of new questions, and that there is likely much more to learn about the role of RNA in plant communication. What we currently know about it is just the tip of the iceberg, she concludes. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.